This video is on membrane liquids. Cell membranes are composed of phospholipids and glycolipids, along with cholesterol for rigidity. Membrane liquids are called amphipathic, which means they have polar and nonpolar components. We will see that they have a polar head and usually two nonpolar tails that go along with it. Here's a table that I'd highly recommend looking at throughout the chapter, and it just breaks down all the different types of membrane liquids, your energy storage liquids, lipids, and just what they all look like. So you'll see that's a lot of pretty tough names to pronounce here at the bottom and the different types. But this is just a table you want to familiarize yourself with. So our membrane lipids, they contain either a glycerol backbone or this is pronounced sphingosine backbone. Your glycerol backbone is what we saw in a previous video. This is your trial from your triacyl glycerols. We saw that before. That was when we took the H off of the alcohols and the OH off the carboxylic acid groups in our esterification reaction. Your sphingosine backbone is a straight chain of 18 carbon atoms. So that's a pretty good indication. If you have 18 unbranched carbon atoms, that's your sphingosine backbone. It also has a trans carbon-to-carbon double bond. It has a hydroxyl group, and it has an amine group. This, I think, really helps looking for that amine group. It's going to be a nitrogen and then maybe like an H2 attached to it. If these lipids have sphingosines, they are called sphingolipids. So A here, this is your glycerol backbone. Again, this is what we saw in the other video. And then here's your sphingosine backbone, your 18 carbons, your amine group, your hydroxyl group, and your alkene. Okay, so going into phospholipids. These are the most common membrane lipid. And it's characterized by a phosphate group. So here is your phosphate group. It's PO4, and then you'll have a double bond to one of the O's. So that is what you're looking for your phospholipids. There are two types of phospholipids. Your first type is glycerophospholipid, and your second type is sphingophospholipid. If you look at your different links of where you're bonded, that can help you determine if it is a glycerophospholipid or a sphingophospholipid. So I want to get into glycerophospholipids here. So these phospholipids are derived from a glycerol compound. So you have to have your glycerol backbone in there, hence the word here, glycero. So here is your backbone. That's the first thing I'm going to look at. Glycerophospholipids will have two nonpolar tails, and these will come from fatty acids. So if you look on the right here, here are your fatty acid nonpolar tails. and then they will come in to join two hydroxyl groups to form your esters. And if you look on page 578 in the book, that is where you will see this depicted as well. Look at the difference now with your sphingophospholippids. Your sphingophospholippids, they are derived from two fatty acids as well, or from a fatty acid linked to an amine group. The other tail is coming from the backbone of sphingo itself. So we know that a sphingosine backbone is a straight chain of 18 carbons. So one of these nonpolar tails is coming from that. So another name for sphingolipids are sphingomyelins, and these insulate nerve tissue in the myelin sheath. You probably have heard of multiple sclerosis before. That is when your myelin sheath deteriorates. Here is something called a glycolipid. So a glycolipid is derived from your sphingosine. So that is why it has this sphingosine backbone here. So you can see your 18 carbons there. You can see your alkene group here, your hydroxyl group, and your amine group there. These are also called glycosphingolipids because they come from your sphingosine backbone. Do not get glyco and glycero confused. Glycero means it has a glycerol backbone. Glyco means there is a carbohydrate component to it. And that's the big difference with these glycolipids. Look at your carbohydrate component on this. That looks like a 5-membered ring. That does not happen in your phospholipids. Let's go back to your phospholipids. Notice there is no carbohydrate component there in either of them. Here you have it. So that's kind of a dead giveaway if you see that it is a glycolipid. There's also not a phosphate group on this. There's no phosphate group. There's no PO4. Again, if we go back, notice here you have your PO4 group, and here you have your PO4 group. You don't have that in glycolipids. If your glycolipid contains a monosaccharide, it's called a cerebroside. These are found in the cells of the central nervous system and the brain. Okay, so here are some questions for us to answer. So the first question says, is it a phospholipid or a glycolipid? So we are looking for either like a PO4 or a carbohydrate group, something like that. I don't see a PO4. I do see a carbohydrate. So because I see a carbohydrate, that means that it is a glycolipid. Does this membrane lipid have a sphingosine or a glycerol backbone? So if it is a sphingosine, we're going to see that 18 carbon chain. We're going to see an alkene, a hydroxyl, and then an amine group. So alkene, hydroxyl, amine group, 18 carbon chain. So this is a sphingosine. I do not see any glycerol backbone there. Is this membrane lipid a sphingophospholipid or glycosphingolipid? Well, it comes from a sphingosine backbone. So I'm going to say sphingophospholipid. And lastly, if this lipid is a cerebroside, identify the carbohydrate and circle the glycosidic linkage. So our carbohydrate is highlighted in yellow, and your glycosidic linkage is right here, from your anomeric carbon to that oxygen. So looking at the cell membrane, your membrane lipids are amphipathic. And remember, that just means it has a nonpolar and polar component. So again, we have our polar head and we have our two nonpolar tails. Your polar head is the spherical notation that you'll see, and then your tails are the longer strands there. If you look at the front of the textbook, you'll see something very similar to this. These are membrane lipids on the cover of the textbook. Your cell membrane has two layers of phospholipids or glycolipids and something called a bilayer. Your polar heads are on the outside. They're in contact with the aqueous environment inside and outside the cell. So you can see here on the surface, you have your polar heads, and then your nonpolar tails are along the inside here together. Okay, we call lipid bilayer fluid mosaic. Important to note that they interact via intermolecular forces. Very weak though, so they're not strong. And you will see different things in the membrane. So here it's saying cholesterol, protein, carbohydrates, which you can see here. Here are your carbohydrates, here are your proteins, the brown, and the cholesterol is the red embedded in there. We learned in a previous video that the permeability of a membrane allows different things in. So depending what's trying to pass through, some can, some can't. We know that small nonpolar molecules can cross through by diffusion, so that's like your oxygen and your CO2. We know osmosis helps water pass through. And then we know anything bigger cannot pass through. So ions, polar organic molecules, large molecules cannot diffuse through or across the membrane. So here are 3 questions. I would encourage you to pause the video and try these on your own, and I will show you the answers.